So I'm just going to talk about some work we've done on these leaf mining flies that are quite invasive pests from around the world. This is work that was conducted in collaboration with DAF, so that's the Australian Government Quarantine Organisation, and through some surveys conducted by NARCS, which is the Northern Australian Quarantine Service. And I'm based at, we're now called DP instead of DPI. Okay, so the Liriomyza sativae is also known as the vegetable leaf miner, and it's a leaf mining fly that predominantly is seen as in the larval or pupal life stages, and they live inside these tunnels inside leaves where they're quite protected from the environment. And Liriomyza sativae is highly polyphagous. It's recorded from 9 plant families. It's quite a serious pest of vegetables worldwide. You can see how much damage it's done to this leaf here with all these tunnels through the leaf. The adults are free-flying, you can see here, but the larvae and pupae occur in these inside the leaves. So it's considered the most serious agromyzid pest, and it's spread a lot through the tropics and subtropics since the 1990s. And it's the dominant leaf mining fly pest in lowland areas of Asia. There's other species that have also spread through the world in this genus, some other serious pests. There's Liriomyza trifoli and Liriomyza hudiborensis, and they're both also highly polyphagous. They occur on lots of hosts. And which species become successful in an area seems to be partly due to insecticide resistance as well as environmental factors. And parasitoid wasps often occur in the pupal stages in the leaves, and they're important in control of this pest as well. You can see it's quite a small fly. It's only a couple of millimeters long as an adult. So they initially came from the Americas, and they've spread through the world pretty much right through, including quite a few areas of the Pacific. And a lot of this spread has happened quite recently. So looking at their worldwide distribution again, they're right through Asia, and then within Pacific part of the world they're also in lots of different islands, but they haven't been recorded from Papua New Guinea or from Australia itself until fairly recently. So the group I worked with on this work was NARCS, which, as I was saying earlier, is the quarantine service that patrols pretty much the tropical border of Australia from about here right round the northern coast to about here. And they have a protected zone in Torres Strait, which is a bit of a buffer between some of the pests that occur up in the north and Australia itself, and then there's a special quarantine zone as these islands that are really close to Australia here. And they conduct surveys in Queensland and also in neighbouring countries. And this is one of my co-authors here on the left. This is Anthony Rice doing a survey in Queensland. And during one of these surveys in 2008, they found some leaf miners in a single tomato plant on an island in Torres Strait, and it turned out that that detection was a Liriomyza sativae, so this serious pest. And since then they've been looking for signs that they've been out there, so they've been looking for these symptoms on leaves, and then we've been helping them with the identifications. So to identify Liriomyza sativae is a little bit difficult morphologically because externally they look very similar, a lot of these species. There's quite a lot of species in the genus and quite a lot of species of agromyzids in general. And to get a positive identification of species, you really have to dissect adult males. So you're limited to just one life stage and one sex for the positive identifications based on morphology. And the stage which is most often encountered is actually the larval or pupal stages inside the leaves. So identifying them from adult males is difficult because you have to raise them through to adults. So molecular identification is very useful in that sort of situation. So for our study, we had samples collected onto these FTA cards, which are basically filter paper cards where you can squish a sample onto them, and it both preserves the sample and starts the DNA extraction process, and they don't need any sort of special temperature treatment or ethanol or freezing, and because they're just paper, they're very easy to transport. So we found these were very effective ways of collecting the larval stages in the field. And for the DNA extraction, you just chop a little bit of the card away and then extract from that. And you can see here there's part of a sample you can just see above where I'm pointing there, and there's a region I've cut away to extract the sample. So a different pupa has been preserved on each of these circles here. There's 4 samples, and then on this card here, a smaller version, we've got 4 samples just squished into one area. And these would all be collected from a single plant. I just point out the disadvantage of this sort of collecting technique is that you're actually destroying the specimen morphologically, so you haven't got that to fall back on. But you could always preserve additional specimens in ethanol if that's what you wanted to lodge some in collections, which is what we've done with our study. So the actual molecular identification, there's lots of techniques that have been used. The most recent that came out last year was to do multiplex PCR, which is where you've got PCR primers that are specific for each species of Liriomyza. And in this case, Liriomyza sativae comes up with a band this size when you use that technique. But in the past, lots of other methods have been used. This looks like a complicated slide, but I'll just show you. It just shows that people have used different sections of these couple of mitochondrial genes here. We've got cytochrome oxidase 1, cytochrome oxidase 2, and initially people amplified up regions of these genes and chopped them with restriction enzymes, which is RFLP, as a way to identify species. And then Sonia Schiefer and her group started doing some DNA sequencing on this part of cytochrome oxidase 1 and looking at populations from around the world. And more recently, other people have started looking at the universal barcoding region, which covers this part of cytochrome oxidase 1, using universal primers, LCO and HCO. And in our study, we looked at that region to tie into this, the Barcode of Life database. There's a lot of species, which I'll show you in a minute, have been sequenced there. And we also developed some new primers to cover this other part of cytochrome oxidase 1. And we specifically designed those primers to avoid potential parasitoids which might be inside the pupae in the leaves. So the data that's out there to compare our results against, there's about 300 species of Agromyzids have been sequenced on the Barcode of Life database, and 26 species of Liriomyza. So there's lots of, once you get a sequence back, you can compare it against all of those and get a identification that way. All of those sequences are tied back to vouchered specimens, and they've been they're a bit more validated than GenBank sequences, for example. And then the other section of cytochrome oxidase 1, we looked at hundreds of individuals have been screened for that particular region by Sonia Schiefer's group. And they've found that there's 3 major groups within Liriomyza sativae, and only one of those groups has spread through the world, this W group. And by looking at that piece of DNA as well, we can tie our data back to that population data and get some idea of where things might have come from. So here's some results from our study. So basically between 2010 and 2012, these leaf miners were turning up from various islands in Torres Strait, which is this region here between Australia and New Guinea. And every individual we sequenced from these different islands, these ones shown in red, turned out to have the same DNA sequence, so the same haplotype was present through all these islands in Torres Strait, and that was between different traditional cultural groups of islands, which is what these white circles are here. So it seemed that Liriomyza sativae has spread over that fairly recent time through a lot of these islands and right down towards the mainland here. Then in 2011, NARCS conducted a survey in the highlands of New Guinea, and we got some samples from 3 villages up here, and it turned out that there were 2 different haplotypes present up in the highlands of New Guinea compared to what we're finding down here in Torres Strait. But we could confirm that that species was present. So Liriomyza sativae is now present, been confirmed from New Guinea and also from the Torres Strait. So another way to view that data is to, these are the New Guinea samples here against, this is a tree of all the sequences that are available on GenBank for this 3' region of cytochrome oxidase 1. And most of the New Guinea samples matched this haplotype here, S7, which is fairly common through the world. And the other sample from New Guinea matched this haplotype here, S28, which again is fairly common. Whereas all the Torres Strait samples were different from these New Guinea ones, and they only matched a single haplotype that has turned up before from Southeast Asia, this S27 haplotype. So basically, we're using DNA barcoding. We were able to validate and verify new location records from New Guinea and Torres Strait from many different hosts, so tomato, pumpkin, and various weeds. It's very useful to use this technique for larval and pupal stages because of the difficulty identifying these things morphologically. And the new primers we've designed to avoid these potential problems with wasp parasitoids within the pupae. It was good to look at a couple of sections of cytochrome oxidase 1 to tie into these 2 data sets, the barcode of life data for the species IDs and then the population haplotype identification, sort of to compare with what's been looked at around the world. We looked at this other section of cytochrome oxidase 1, and that showed these that the Torres Strait introduction seems to be different from the New Guinea introduction. But what we haven't done yet is screen surrounding areas, so we haven't looked at any samples from the various islands in the Pacific or even from Indonesia and other parts of Southeast Asia. So that would be a useful addition on top of this study. And we also haven't started looking at using pinned specimens from collections, which would be good too. The final point I'd just like to make is that we haven't detected Liriomyza sativae from the mainland of Australia yet. It's only from this region of Torres Strait and New Guinea. And that's it. Thanks.
Thanks very much, Mark. I'll try to minimize my time on microphone since I know there's an echo. We're waiting for questions to come in. We have one follow-up question for Bevan. In the meantime, Bevan, if you unmute your microphone, Diego clarified his question. He said, by reevaluating, are you going back into the field or using specimens in the collection to reevaluate identifications?
Yeah, often it is going back into the specimens in the collection because, you know, they're right there. For me, it's just a few meters on the other side of the corridor, and I can take them downstairs and sequence them. Whereas going out into the field, you know, like I said before, you know, you have to find the disease and you have to potentially be at the right season. So looking at the collections is always the first port of call. But looking at new specimens is also really important. because you don't know, is the organism that caused the disease 30 years ago the same organism that's causing the disease today? And often it's not. And you can have seasonal and variations from years, and also geographic variation as well.
I'm sorry, did that come through? The question was, have you considered using high throughput next generation sequencing? Is that directed to Mark or to... Okay, Mark or Bevan, could you comment on your potential use of next gen?
I'll say something first. I think for the particular identification work we were doing, I think that doing the mitochondrial sequencing is enough just to get the identification, even to tie them back to population. So there's no need to go to next generation sequencing for the work we were doing.
Yeah, I agree. Next generation sequencing is great, and we've used it for some environmental applications. But in a plant, it's normally only one organism causing that disease. And so, often you can the best thing is just to pick it off or to isolate it off. One thing that we are looking at next generation sequencing is to go back and sequence some of these really old herbarium records. And because next generation sequencing can, particularly Illumina, can pick up very, very small fragments and piece them together, we're hoping that we can even get degraded DNA and sequence that and get a workable barcode.
Yeah, certainly CBOL has been watching the development of next generation, and except for environmental mixtures, it really doesn't seem to make economic sense yet because of the cost of sample preparation and informatics. I do have a question for Mark. I just wanted to be clear that the 3' end that had more population level information, were you able to do that in one run with the barcode region, or did you use, it sounded like you were using internal primers for separate sequencing runs. Did I get that correct?
Yeah, that's right. So it was one PCR reaction, but sorry, it was using one PCR reaction in the machine, but we're actually, you're right, there's a set of primers for the 5' and a set of primers for the 3', and we'd sequence those two separately and then just analyze them separately. So they could be amplified using the same annealing temperatures and all that sort of stuff, but they were looked at separately, just instead of looking at the whole of CO1 together. I guess that would be another option, just amplify the whole thing up.
Do you think you could do the entire sequence in one go on the sequencer and then divide it informatically later?
Yeah, I think that would definitely be a possibility. But I mean, we were specifically using these new primers to avoid potential Hymenopteran parasitoids. So that was one reason to break it up into those two sections.
That is a clever strategy. For Mark, here's a question from Saloti. I'm not sure if I'm pronouncing that right. How accurate have your identifications been between the genetics and morphological data from adult males?
Yeah, we haven't done a lot of that validation. It would be really useful say take a single leg or something like that off specimens that have been pinned and confirm those IDs, but the actual DNA IDs are very, there's quite a lot of, all this background information has been done on the DNA side of things that we're very confident of the IDs.